Hi everyone, this is the first of a three-part series on the immune system. So what it does is it provides resistance to disease. And so we have two intrinsic systems, so two internal systems in the immune system. We have the innate or the nonspecific defense system that is in place when you were born. You're born with this. You don't have to do anything to get this. It's just there. It takes up what we call the first and second lines of defense. The first line of defense is the external body membranes, basically the skin and the mucosa. And so that's what's keeping the outside world out and the inside world in, and it's protecting you from what's in the outside world. If something manages to get through that particular line of defense, then we have the second line of defense. That is the antimicrobial proteins, phagocytic cells, and other cells that are able to inhibit the spread of invaders, inflammation, some other types of mechanisms that we have to help keep the microbes or pathogens to get from getting any further into our system. And then we have the adaptive or the specific defense system, which serves as our third line of defense if something manages to get past the first and second line of defense. And so the third line is going to attack against a particular foreign substance, an antigen, and it's going to take longer for it to react. This is the side of the immune system that you're building when you go through the process of getting sick and all that sort of stuff, or you get immunizations, all that sort of thing. So the immune system is a functional system rather than an organ system. All of the structures that we talked about with the lymphatic system, those are the structures, if you will, that are housing the cells, but this is actually a functional system. So we talk about the innate and the adaptive defenses separately, but in reality, they are actually intertwined. They're working together. Both are able to release and recognize a lot of the same defensive molecules. The innate defense system does not have specific pathways for certain substances, but the innate will. The adaptive will. The innate response also is going to release proteins that alert cells of the adaptive, so that's how they're working together. So here is what I was just talking about in the very beginning, if you want to see it in a slide, in a picture form. So our innate defenses, again, innate means that you were born with it. It was available to you. It was part of the natal period, or when you're born, right? So it's already in place. So that's going to be your surface barriers like the skin and the mucous membranes, and those are considered the first line of defense. Then you have what's right underneath of the skin and some other internal defenses that are going to try to limit the spread of infection should it make it past the skin and the mucous membranes. These are going to include the phagocytic cells, natural killer cells, inflammation, antimicrobial proteins, and fever. Right, so those are all going to be things that you're born with. You don't have to do anything to get these. They're going to act the same every single time, but we have them in place as our second line of defense. Then if something manages to get past all of that, we have our adaptive defenses, which is going to be is the immune system that we actually have to develop over time. And so this is going to be further divided into the humoral immunity, which is going to be carried out by the B cells, and the cellular immunity, which is going to be carried out by T cells. So let's talk about the innate defenses first. And a lot of people, when they first hear this, they thought, well, I never really thought of that being part of my immune system before. But actually, it really is. So your innate system uses its first and or second lines of defense. And what it's trying to do is stop the invasion of pathogens. So pathogens are any microorganism, whether it be a virus or a bacteria or a protozoan, fungi, whatever it is that can actually cause disease. So let's talk about your surface barriers first. So skin and mucous membranes are considered surface barriers along with the secretions that they make. They provide a physical barrier to most microorganisms, so fully intact skin and mucous membranes mean that things can't get through that. Keratin is resistant to weak acids and bases, bacterial enzymes, and toxins. So we don't have any concern about those particular things being able to get through because the skin has a lot of keratin in it. The mucosa provide a similar mechanical barriers. These are the surface linings of the cavities that open to the outside world. Skin and mucous membranes produce chemicals that inhibit or destroy microorganisms. One thing that they produce is acid. So the natural acid mantle, the acidity of your skin, and some of your mucus secretions help to inhibit growth. So that's what keeps bacteria from being able to grow on your skin. The unfortunate side effect of that is that Fungi, unfortunately, like slightly acidic environments. So fungi grow easily on the skin and mucous membranes, which is why in unbroken skin, you typically don't see a bacterial infection, you see a fungal infection. So things like um, athlete's foot and jock itch and ringworm, all of those kinds of things. Thrush, which is the infection that gets in the throat of the baby. Those are uh, yeast infections in women. Those are all fungi, and they like the slightly acidic environment that we produce for them. Enzymes, so that's another thing that can kill bacteria. This is found in the lysosome of saliva, in the respiratory mucus, lacrimal fluids. They all help to kill these microorganisms that we come in contact with. The enzymes in the stomach has a pH that's really, really low that helps to kill the microorganisms that are in the food and water and things that we consume. Mucin, which is a sticky mucus that lines the digestive and respiratory tracts, helps to track microorganisms, and so we spit that out, cough that out, or swallow that down into our stomach when it's in our head area. Defensins are antimicrobial peptides that inhibit the microbial growth. And then there's other chemicals like the lipids that are in our oil or body oils or sebum, and the dermcidin in our sweat. These are all toxic to certain bacteria that get on our skin. The respiratory system has some special modifications that help to stop pathogens. You have mucus-coated hairs that are trapping inhaled particles in the nasal cavity. You have cilia in the upper respiratory tract that help to sweep the dust and the bacteria-laden mucus into the mouth. They call that the mucus escalator. You have surface barriers that are breached by nicks and cuts. This is actually going to mean that the first line of defense is no longer in place, so that the second line has to be activated to protect the deeper tissues. So just taking a look at the first line of defense, the surface membrane barriers in a nice chart form for you, giving you an idea of what all of the different first line of defenses are and how they work to help us and protect us. Then we have the second line. These are cells and chemicals. Again, these are part of our innate immune system. This is necessary if microorganisms manage to get into deeper tissue. So this is going to include phagocytic cells, natural killer cells, the inflammatory response, which is going to include macrophages, mast cells, white blood cells, and inflammatory chemicals, antimicrobial proteins like interferons and complement proteins, and fever. So many second line cells have pattern recognition receptors that help them to recognize and bind tightly to things, structures on microbes. In other words, they have the ability to recognize foreign cells, and that helps to disarm them before they do any kind of damage. So let's talk about your phagocytic cells. These are white blood cells that ingest and digest or eat foreign invaders. Neutrophils are the most abundant type of these. We learned these in our blood chapter. So remember, neutrophils are the most common white blood cell, the most abundant that you have. Never let monkeys eat bananas. So neutrophils are for never. These are the most abundant, but they die really quickly because they are involved in hand-on-hand combat. They become phagocytic on exposure to infectious material. Macrophages develop from monocytes. These are our chief phagocytic cells. They are the most robust phagocytic cell that we have. There's two versions of them. We have the free macrophages that wander through the tissue spaces. So a good example of these would be the alveolar macrophages that are found in the lungs, also known as the dust cells. The fixed macrophages, these live permanently in some organ. So we have the stellate macrophages, for example, in the liver, or the microglia that we saw last semester in the brain. And so here is just a picture of a macrophage, the big purple guy, and he's using his cytoplasmic extensions to pick up those little green rod-shaped bacteria to ingest them. So phagocytosis, what is the process of this? So it starts when the phagocyte, the white blood cell, recognizes and adheres to a pathogen's carbohydrate signature. So if you want to think about it like this, all cells, whether they belong to you or don't belong to you, have a flag on them. The cells that belong to you have a flag that says, I belong. The cells that don't belong to you, whether it's a bacteria or other type of cell that gets in your body, or a virus virus or whatever, they have a flag that says I don't belong on them, and so that flag is actually a carbohydrate signature. Some microorganisms have external capsules that hide those flags, and it helps them to evade phagocytosis. Opsonization is a process that the immune system uses with antibodies or complement proteins that coat the pathogen, and essentially it makes little handles for the phagocytes to grab onto. I always refer to it as like sugar coating. So you put the sugar is sticky, you put it on the outside of whatever it is that you want engulfed, and it makes it more attractive to the phagocytic cells. So there they're showing you the events of phagocytosis. The phagocyte adheres to the pathogen or the debris, and then the phagocytic extensions or the pseudobonds are going to bind and engulf that particle and create something called a phagosome. The phagosome is going to fuse with a lysosome, which forms a phagolysosomes, and the phagolysosome is acidified. The lysosomal enzymes are going to digest the particles, and the indigestible and residual waste is exocytosed from the phagosome. So there's the rest of this. So we started with the little green things are your, let's just call them E. coli, because green E. coli actually comes up metallic green if you stain it properly in microbiology, and they are rod-shaped like that. So we're just going to pretend that those are little E. colis. So you see what happens is the big blue phagocytic cell adheres to the pathogen, the little E. coli. The phagocyte forms pseudopods, little false feet that engulf and bring that that little vesicle into the cell. So now it is a phagosome, which means a phagocytic vesicle full of stuff we engulf. And then lysosome is going to fuse with that. Remember, lysosome was the organelle that's responsible for secreting digestive enzymes. And so it forms a phagolysosomes when those two fuse together. And then the toxic compound and the lysosomal enzymes are going to destroy the pathogens and basically neutralize them. And then they're going to be exocytosed from the cell. So that's how a white blood cell engulfs and kills something. Some pathogens are not killed with acidified lysosomal enzymes. Tuberculosis pathogen is a good example of this. Helper T cells are going to help with this process by triggering the macrophage to produce a respiratory burst, which kills the pathogens that are resistant to these enzymes. It's almost like the macrophage sneezes, if you will, onto the pathogen, and that causes the releasing of cell-killing free radicals. They produce oxidizing chemicals like H2O2, which is hydrogen peroxide, and that increases the pH and osmolarity of the phagolysosomes. So it's just another way of killing resistant, if you will, bacteria. Defensins that are found in neutrophils also help by punching holes or piercing the membrane of a pathogen. So we'll see that a lot of our white blood cells act like little switchblade carrying ninjas, in the sense that they have the ability to punch holes in the cell membrane of the offending cell, and when you have holes in your cell membrane, you die. What about your natural killer cells? These are non-phagocytic cells, large granular lymphocytes that police the blood and lymph, and they can kill cancer cells, virally infected cells, before the adaptive immune system is ever activated. Remember, that's the last line of defense. They attack cells that don't have that surface membrane that indicates that it belongs to the body, so it doesn't have the right flag. They kill by inducing apoptosis, in other words, programmed cell death in cancer cells and virally infected cells. They secrete potent chemicals that enhance the inflammatory response as well. So let's talk about inflammation for just a second. Most of the time, we think about inflammation as a bad thing, but inflammation actually has a function in the body. It's when it spirals out of control that it becomes a bad thing. But inflammation is triggered whenever you have injury to body tissues. That can be because of trauma, like you sprained your ankle, or a burn like heat, or an irritating chemical that you've come in contact with, or an infection by a microbe. The benefits of inflammation include preventing the spread of damaging agents. So that's kind of the idea. We want to inflame that area and keep things from leaving that area. It also helps to dispose of cell debris and pathogens. It alerts the adaptive immune system, and it sets the stage for repair. There are 4 cardinal signs of acute inflammation. They include redness, heat, swelling, and pain. There is often a 5th sign that we use for that criteria of acute inflammation, and that's impairment of function. So that particular area can't do what it did before, especially if you're talking about movement. So the stages of inflammation. So you have an inflammatory chemical release, you have vasodilation and increased vascular permeability, and you have phagocyte mobilization. So the inflammatory chemical release happens when chemicals are released into the extracellular fluid by tissues that are damaged, also released by immune cells or blood proteins. So for example, histamine is released by mast cells, and this is the key inflammatory chemical. So the macrophages and the epithelial cells of the boundary tissues, like respiratory tissues and the intestine, they bear a special recognition receptor called a toll-like receptor, or a TLR. There are 11 types of TLRs that recognize specific classes of infecting microbes. Activated activated TLRs trigger release of the cytokines that promote inflammation. Some other inflammatory mediators besides histamine, you can have kinins, you can have prostaglandins, complement. All of these work kind of the same way. They all cause vasodilation in the local arterioles, so they cause them to enlarge and make the capillaries very leaky. When that leakiness happens, it attracts leukocytes to the area. Some have other inflammatory roles, like triggering pain receptors or prompting the release of more inflammatory chemicals. So here is a table 20.2, of the inflammatory chemicals that I was just talking about in a little bit more detail, so you can see how they work. So again, we're still talking about inflammation and the tissue injury response to injury. So vasodilation and increased vascular permeability, this causes hyperemia or congestion in the area with blood, which leads to the redness and the heat that we see related to inflammation. Increased capillary permeability causes extrudent or fluid containing clotting factors and antibodies to leak into the tissues, and that results in local swelling or edema. The swelling pushes on the nerve endings, and that's what causes pain. The pain can also be a result of the release of toxins from the bacteria or any released prostaglandins or kinins. There are benefits of edema. A surge of fluid into the tissue is going to sweep out anything foreign that has gotten into the tissue and push it into the lymphatic vessels so that it can be processed in the lymphatic nodes. It also delivers clotting proteins and complement to the area. The clotting factors are going to form the fibrin mesh that we talked about back in the blood chapter. That's going to start as begin the scaffolding for repair. And that mesh is also going to isolate the injured area so invaders can't spread. So anything else that continues to come into that area can't get loose into the rest of the body. Phagocyte mobilization, neutrophils again are always the first to arrive. The macrophages are going to follow those. If the inflammation is due to pathogens, complement is going to be activated and the adaptive immunity elements will begin to arrive. So what are the steps for phagocyte mobilization? Leukocytosis is the release of the neutrophils from the bone marrow in response to leukocytosis inducing factors from injured cells. So the injured cells release a chemical called LIF, and when they release that, it causes the bone marrow to release extra neutrophils. Margination is when the endothelial cells in the capillaries in the inflamed area project cell adhesion molecules or CAMs into the lumen of the vesicle, and they grab those neutrophils as they float by, causing them to slow down and cling to the vessel wall. Then that neutrophil is going to squeeze through between the cells of the vessel wall and move into the interstitial spaces. That process is called diapedesis. It basically means to creep using feet, which is kind of what it looks like. And then we have chemotaxis that occurs. So we have the neutrophils that have gotten out into the tissue, but how do they know where to go? So we have to make sure they know where to go, and inflammatory chemicals are being released by the injured cells, and those chemicals are like the Krispy Kreme sign in Charleston when the hot donuts are ready, right? It tracks everybody right there. And so how does a little teeny tiny white blood cell know where to go in a great big huge body? It looks for the Krispy Kreme sign. It looks for the chemotaxis sign that's being released by the neutrophils, the chemicals that are being released, and that's what attracts it to its area. Some people say it's like it smells its way there, so I don't want you to walk away thinking that white blood cells have the ability to smell anything. They're just attracted by chemicals. As the attack continues, as the monocytes are going to arrive. 12 hours after leaving the bloodstream, they are transformed into macrophages. So as you will remember, monocytes become macrophages when they're in the tissue. These late arrivers are going to replace the neutrophils that are dying from the assault and remain for the cleanup prior to the repair. All right, so showing here, you hear how the phagocytes are mobilized. So this is going to be using our innate defenses, which are going to involve our internal defenses. So you managed to step on a nail or a tack or whatever that is, and you've broken the skin, and it's gone through to the dermis, and it's released some of those little E. coli's in. And so what's happening down at the blood supplies? Well, leukocytosis is happening. The neutrophils are entering the blood from bone marrow because of the release of the chemicals by the damaged cells where the nail went in. Margination is happening. The neutrophils are clinging to the capillary wall. Then they're squeezing out through diapedesis. And they're heading over through chemotaxis. They're following their nose over to where the chemicals are, where the dam is, and they're going to start picking up those green things. So this chart is telling you kind of a flow chart of all the different things that are going on with the innate defenses whenever you have some type of an injury and what all's going on. So you can kind of follow that through so you can see all of the different events that are happening and what the different events are causing the cardinal signs of inflammation. So then we have a little homeostatic balance to talk about. It's called pus. So I have a lecture on here that's not recorded that says fun stuff to know, and it's the different types of pus. Pus actually can be really diagnostic, believe it or not, so you can look through that in your own time. I'm going to cover it in the lecture, but I don't record it, so you can take a look at that as well. So what pus is a mixture of dead neutrophils that died in the battle, tissues and cells, and pathogens that are either living, still alive, or ones that have already been killed. Okay, so that's what pus is. So pus is kind of an indicator that your immune system is doing what it's trying to do or it's supposed to be doing. An abscess is when collagen fibers are laid down and they wall off the pus, and that we have to surgically drain. So if that happens, you get an abscess, they have to drain that because you're not going to be able to get rid of it any other way. Some bacteria, like the tuberculosis bacilli, are able to resist digestion by the macrophages, and they can remain alive inside. They can form these tumor-like growths called granulomas. These are areas of infected macrophages that are surrounded by uninfected macrophages and outer capsule, and that bacteria may remain inactive forever, or if the person's immune system crashes or decreases for some reason, they can break free and become activated and cause the disease. Antimicrobial proteins, these help to enhance the innate side of our immune system by either attacking the microorganism directly or hindering the microorganism's ability to make copies of itself or to reproduce. The most important antimicrobial proteins are the interferons and the complement proteins. So interferons, or IFN, is a family of immune modulating proteins. These cells are kind of like the town crier, whereas they warn the rest of the cells in the population that a virus has invaded them. And so when a cell has been invaded by a virus, it secretes interferons that warn all of the neighboring healthy cells. So the interferons enter the neighboring cells. They stimulate the production of proteins that will block the replication of the virus and degrade viral RNA. There's two different types that work this way, alpha and beta interferon, and they also work to activate natural killer cells. Another type of interferon is called gamma. So this is called immune interferon, and it's secreted by the lymphocytes. It has widespread immune mobilizing effects, and it activates macrophages. Interferons activate the natural killer cells and the macrophages, so they indirectly are in the process of fighting cancer. We can use artificial interferons to treat disorders like hepatitis C, genital warts, and multiple sclerosis. And so here it's showing you the interferon mechanism against a virus. So basically it warns, it's a chemical that warns the other cells that, hey, I've become infected. You don't want to let this virus attach to you and infect you as well. Showing you how that happens in the different steps. Complement is a system of approximately 20 blood proteins that circulate in the inactive form in our blood. They include proteins C1 through C9, factors B, D, and P, and some regulatory proteins. They provide the major mechanism for destroying foreign substances. And so complement, its name kind of tells you what it does. It enhances or it complements inflammation and also directly destroys bacteria. So essentially what complement does is it helps the innate immune system and the adaptive immune system do its job better. So it complements it. Not like compliment like saying something nice, but compliment like helping. All right, so that's why it's spelled differently. So the complement system has three different pathways of activation. The classical pathway, antibodies are first going to bind to the invading organism, and then they're going to bind to the complement components and activate them. This double binding is called complement fixation, and once the initial complement proteins are activated, a cascade is triggered. The lectin pathway, lectins are produced by the innate system to recognize foreign invaders. When the lectin is bound to a specific sugar on a foreign invader, or in other words, it's flagged that it's flying that's not supposed to be there, it can also bind and activate complement. And then the alternative pathway, this is complement cascade that's activated spontaneously when certain complement factors bind directly to the foreign invader. And so these lack the inhibitors on the microorganism surface to allow the process to proceed. Each pathway involves an activation of proteins in an orderly sequence, and that's the important idea out of this. It doesn't matter which of these three ways it happens, each step catalyzes the next, so one right after another, like dominoes, one right after another. Each pathway ultimately is going to converge on C3, which cleaves into C3A and C3B, and the splitting of C3 initiates a common terminal pathway that's going to enhance inflammation, promotes phagocytosis, and causes cell lysis. So regardless of the way that it's triggered, at the end of the game, it's all going to end up doing the same thing, and that's to help the innate immune system do its job better. Cell lysis begins when the C3b binds to the target cell. It triggers the insertion of a complement protein called the membrane attack complex, or the MAC, into the cell membrane. So we call this MAC attack. MAC forms and stabilizes a hole in the membrane and causes the influx of water and causes the cell to rupture. So basically it makes a hole in it, and it's almost like it sticks a garden hose in there and blows up the cell. C3b also causes optimization or coating, sugar coating. C3a and other complement proteins cleave products that amplify inflammation by stimulating the mast cells and the basophils to release histamine. They also attract neutrophils and other inflammatory chemicals. So there's the idea. Again, classical, lectin, and alternative. You want to know those three terms and what they mean, and know that they all merge on C3, and it's C3 depending on whether it goes C3b or C3a. It can cause opsonization, it can cause a MAC attack, it can cause enhancing of the inflammation, so all different types of things that happen. Another one that you might not have thought of as being actually a part of your innate immune system is fever. So an abnormally high body temperature is a response to pathogens, microorganisms that have come in. Leukocytes and macrophages exposed to foreign substance secrete a chemical called a pyrogen. Pyrogens act on your body's thermostat in your hypothalamus, and they cause you to crank up the temperature just a bit. So a moderate fever, moderate meaning under 100.3, not 103, but 100.3. That's considered moderate fever. That causes the liver and the spleen to sequester the iron and zinc, and microorganisms must get iron and zinc from your body to continue to divide. And this is going to increase the metabolic rate, which increases the rate of repair. So the idea is that we basically turn the temperature up just a little tiny bit, and that makes the microbes not very happy, and they can't divide, and they go away. And finally, a table that reviews the second line of defense, all the things we were just talking about. A really good way to review and make sure that you have all of the information. I'll see you for the next part.